Antibacterials are commonly known as antibiotics, and antibiotics are molecules that prevent growth and multiplication of bacteria. People discovered bacteria in the 1600s, but it wasn't actually until about the 1860s that we began to connect bacteria with human diseases. And so it wasn't until about that time, the 1860s, that people really began to look at ways to inhibit bacterial growth in an effort to cure or to treat human disease. And so 1908 is really the first time that drugs were intentionally given to treat a bacterial disease, and the bacterial disease was actually syphilis. And then in 1927, so 60 good years after we first began to connect bacteria with human disease, a guy named Twite published a paper indicating that the fungus Penicillium inhibited growth of bacteria. And I think that the story goes that this guy was trying to grow some bacteria on some plates, and he left the plates by an open window, and some fungus blew in from the open window and landed in the plates, and he noticed that everywhere that the fungus grew, the bacteria did not. And so that began a search for what was inside the fungus that inhibited the growth of the bacteria. 2 years later, a man named Alexander Fleming published another paper in the scientific literature suggesting that the fungus Penicillium actually had a molecule inside it that he called penicillin that he said was the molecule that inhibited bacterial growth. Also in 1935, a sulfa drug called protonsil was used to treat strep for the first time. 11 years after penicillin was first discovered, so in 1941, it was finally used to treat a person. So the person was a policeman who had obtained an injury, and he temporarily got better. But the problem was that the people who gave him the drug didn't have enough of it to completely cure him, so to completely kill off all of the bacteria. And so eventually the bacterial populations in his body increased enough that he became sick again, and he actually later died. But that encouraged people to figure out how to mass produce penicillin because people realized that it would be useful for treating a wide variety of bacterial diseases. In the 1950s, we finally figured out what the molecular structure of penicillin was, so what the molecule looked like. And the advantage of knowing what it looked like was that we could figure out how to make it in bulk, so we could synthesize it in chemical laboratories rather than relying on the fungus to make the chemical. The other advantage of knowing what the molecular structure looked like was then that we could tweak the structure slightly to create new antibiotics which were effective on different bacterial strains or were stronger and more effective than penicillin itself. The picture at the bottom of this page shows a diagram of a bacterial cell. We don't want to stray too near biology here because, far be it from me to teach you any biology, but the important part of this picture is actually the outside of the bacteria and not what's inside. So the thing that you need to know about a bacteria is that it is surrounded by a cell wall, and the cell wall sort of holds in the guts or the inside material of the bacterial cell. And so we can think of the cell wall as kind of the sac in which the insides of the bacteria exist. So penicillin works by actually interfering with enzymes that the bacteria need to make their cell walls. And what happens to the bacteria when their cell walls don't work is that they swell, and eventually the cell wall will burst. For quite a while, penicillin was the sort of go-to antibiotic for the treatment of bacterial illnesses. However, we've recognized that over time, some bacterial strains have become resistant to penicillin, and so the penicillin isn't effective at killing those bacteria strains. Those particular strains contain an enzyme called penicillinase, and we named it that. And the purpose of penicillinase is to deactivate penicillin. Uh, One of the primary reasons that bacterial strains have been able to develop resistance is that antibiotics are used abundantly, sometimes just to treat minor illnesses, but actually one of its primary uses is not in humans, but in animal feedstock. So we have these images in our heads of cows grazing in wide open fields, and in reality this isn't how most cows that we eat today are raised. They're raised on feedlots, so imagine essentially a parking lot that's paved, so no grass, and with a fence around it, and many cows are put very close to each other, and they eat and drink from troughs. And when people are really close to each other, they tend to share illnesses more readily, and cows are the same way. So when cows are in close proximity on these feedlots, they tend to get sick more often. And so animals, cows, chickens, pigs, whatever, they're all kind of treated the same way. When they're in close proximity like this, they're administered pretty large doses of antibiotics to keep them from infecting each other with disease. And unfortunately, the use of antibiotics in livestock is one of the reasons that bacteria have developed a resistance to penicillin. When a person gets a disease, some sort of resistant bacterial strain, that person will often have to take a cocktail of several antibiotics, and some of them can be penicillin derivatives, and some of them might be other antibiotics that have been developed since penicillin was developed, and the patient has to follow a really strict treatment regimen, or the bacterial populations may actually get worse instead of better. In just a moment, we'll be looking at the chemical structure of penicillin, but here are some of the highlights of what penicillin looks like. So all penicillin derivatives have a four-membered ring containing a nitrogen, and that ring is called a beta-lactam ring. Again, it's common not just to penicillin, but all penicillin derivatives, so newer antibiotics, derived from penicillin as well. Another common feature to penicillin derivatives is that they all have a side chain that comes off of that four-membered ring. And what's different about different penicillin derivatives is the actual chemical makeup of that side chain. The nice thing about the side chain is that it can actually be altered slightly so that that bacterial enzyme penicillinase that actually deactivates penicillin, if we change the side chain, then the penicillinase will not actually work. And so newer derivatives of penicillin are less susceptible to bacterial resistance at the moment because penicillinase doesn't work on them. Another thing that can be altered in the side chain is that we can make the molecule less susceptible to breakdown in the stomach due to contact with hydrochloric acid, so stomach acid. So originally penicillin had to be taken by injection or parenterally, because if it wasn't given by injection, it wouldn't make it through the digestive system into the bloodstream without being broken down by stomach acid. So by altering that side chain, we make penicillin less susceptible to degradation by hydrochloric acid, and now we have the joy of being able to take our penicillin or antibiotics usually orally rather than having to take them by injection. Antibiotics are sometimes given by injection still, but typically if they're given by injection today, the purpose of that is to get them into the system faster. Here you see the general structure of all penicillin derivatives. This little four-membered ring here with the nitrogen on one corner is the beta-lactam ring, and this part here is the side chain. And notice there's a little R, and we think rest of the molecule, like there's some more to the chemical structure that we just didn't draw. That little R group is what changes in the side chain to make different penicillin derivatives. So different penicillin derivatives have slightly different chemical structures, but they all have a side chain of some variety and this beta-lactam ring. And then finally, just to reiterate the problems associated with overuse of antibacterials or antibiotics, the first problem is that if antibiotics are prescribed to people or to animals too often, bacteria develop resistance to them. And so we have to continuously develop stronger and newer antibacterials to combat resistant strains. And then the other problem is that antibiotics aren't selective, and so they don't kill only bacteria that cause human disease. They kill pretty much all bacteria, and so there are actually multiple bacterial strains within people's bodies that are actually useful rather than harmful. And in fact, if we don't have these bacterial strains in our bodies, we can die. Of particular interest, there are bacterial strains in the digestive system, so in the intestinal tract, that serve some very important functions in the human body. And the problem with using antibiotics frequently is that they can kill those useful bacteria in the digestive tract, and you can actually die from having those populations killed off in your own body. Now we often prescribe probiotics with antibiotics. So a probiotic is actually a bacterial culture, so a bunch of bacteria that you swallow, and the bacteria that you swallow are good bacteria, so not harmful ones, and they go into your digestive tract and help repopulate your digestive tract with useful bacteria when you're done taking the antibiotics to kill off the bacteria that were hurting you.